Hello, welcome to the infectious diseases. And today we are going to speak about diseases with vector-borne mechanism of transmission. Especially we will speak about the malaria, sure, because it's one of the most often parasitic infection all over the world and still today very dangerous and can lead to death. But for the beginning, the general characteristic of blood infections. You have to understand what does it mean blood infections and what are the main peculiarities of such infections. Infections included in this group are integrated by vector, transmissive mechanism of infection. Transmission is conducted by a live transmitter. Can be any, but as a rule arthropods, you have to remember. From the blood of infected microorganism into the organism of transmitter, the agent enters during blood sucking. At the repeated blood sucking on the other microorganism occurs the transmission of the agent into the new host. The most of blood infections are typical zoonoses. Anthraponosis, as a rule, yes, that is the most dangerous for humans. As a rule, malaria, parasitogenic typhus, like epidemic and endemic loose infected typhus, trench fever, and some helminthiasis. Transmitters of blood infections, what can be? As a rule, arthropods, you see, but flea, lice, ticks, mosquitoes, any. I hope you remember, yes, tsetse fly, interesting name, sand flies, and so on. The transmitters can be only the arthropods. In the organism of what the parasite can replicate or perform some cycles of development. Only mechanical displacement can guarantee the delivery of agent in the infected doses. That's why remember, as a rule, the agent has to stay and leave a little inside the transmitter. Without such process, the doses maybe will not be enough. The arthropods are infected at blood sucking of the sick animal or human. The agents replicate in the salivary glands, malaria, tick encephalitis during, or in the digestive tract during plague, epidemic typhus, trench fever of exoparasites. At the next blood sucking, the agents will, with the saliva, hemolymph, spit up masses, or excrements are brought into the susceptible organism. Out here you can see like epidemiological moments, the transmissive mechanism or vector-borne mechanism in other words, but I want you to remember also possible such name and such acts of this process. The first is the source of infection. I hope you remember from the beginning of epidemiology, yes, the epidemiological process. First is the source of infection. The agent is situated in the blood system, like closed system, and only by the vector it can be transmoved from this closed system and by the environment, but exactly inside the transmitter, inside the vector, can penetrate, yes, in the susceptible organisms, the third chain of epidemiological process. Penetration into the blood of susceptible organism. The main peculiarities of blood infections, what you can see, endemicity. It's understanding, yes, because exactly for such regions where the transmitter live will be characterized such diseases. Registration on the territories where the transmitter live. Seasonality. also time when these transmitters are living or replicating or some another processes. The contamination occur in the period of transmitter's activity. At zoogenic infections, the human, as a rule, is the biological dead end. Remember. And now we will start more close and very carefully to discuss about malaria, because malaria still is one of the most spreading in tropical areas and most devastating, most frequent, most serious, severe disease in tropical regions. Yes, exactly tropical, because a mosquito, an Anopheles mosquito, can live just in such regions when the average temperature is about 16, higher than 16, not lower. Just one plasmodium can live during lower temperature. Malaria. Name is derived from Latin mal area or bad air. You understand? Yes. What's this miasmas? Maybe you remember from the previous lessons. Lectures, I told you about such a word miasma, like bad air. Because in the ancient and medieval also times, a lot of scientists thought that exactly bad air caused the diseases. And malaria had at that time was, yes, better to say was, the most often disease, and exactly for such disease was used such name. Malaria continues to be the most important cause of fever and morbidity in the tropical world. Malaria has been eradicated from Europe, most of North America, Korea, and Japan. Here you can see a lot of different famous persons of our history, humanity history. Malaria has always been and is one of the most dangerous human diseases. It led to the decline of large countries, destroyed peoples, decided the outcome of wars. Famous victims most likely to have died of malaria include Alexander the Great, you see, Alaric, king of the Goths, Attila, king of the Huns, Genghis Khan, you see, yes, Saint Augustine, the Italian poet Dante, the Holy Roman Emperor Karl V, Christopher Columbus, Oliver Cromwell, Lord Byron, and many others. Here you can see the malaria endemic areas, and you see, as a rule, a so-called malarial belt is present in our planet. What you see is the tropical all regions involved in this process. And here you can see the cases and maybe the zero cases from the 17th. Malaria remains the world's most devastating human parasitic infection. Malaria affects over 40% of the world's population. Can you imagine? It's like about COVID already. World Health Organization estimates that there are 500 million cases of malaria worldwide, of which 400 million are falciparum malaria, the most severe form of the disease. Malaria kills more people than AIDS. Malaria kills in 1 year what AIDS killed in 15 years. For every death due to HIV, there are about 50 deaths due to malaria. To add to the problem is the increase in drug resistance to the established drugs. Some words about history. Historical points. You understand already that malaria was known from the ancient and and even prehistorical times. The symptoms of malaria were described in the ancient Chinese medical writings. In 2700 before Christ, several characteristic symptoms of what would later be named malaria were described in the Huangji Neijing, the inner canon of the Yellow Emperor. Sure, Hippocrates, born, no, you know, yes, who is Hippocrates, father of medicine, was the first to describe the manifestation of the disease and relate them to the time of year and to where the patient lived. And already scientific approaches in the malaria. Charles Louis Alphonse Laveran, a French army surgeon stationed in Constantine, Algeria, was the first to notice parasites in the blood of a patient suffering from malaria. And sure, he was the first. This occurred on the 6th of November 1880. For his discovery, Laveran was awarded the Nobel Prize in 1907. He first mentioned the presence of parasites. Ronald Ross, don't forget about such person. In August 20, 1897, Ronald Ross, a British officer in the Indian Medical Service, was the first to demonstrate that malaria parasites could be transmitted from infected patients to mosquitoes. Because, yes, parasites present in blood, Alphonse Laveran, yes, proved this, that parasites present, but how they appear in this blood, nobody can understand. That's why, what exactly, due to Ronald Ross, the scientists, the doctors understand that this disease and these parasites can be transmitted exactly by mosquitoes. For his discovery, Ross was awarded by the Nobel Prize in 1902, even earlier, you see, than Alphonse Laveran. Because the transmission was even more interesting and important, and now also because the prevention, the understanding of transmission will understand the possibility of prevention, will lead to understanding. Malaria, yes, sure, vector-borne disease, you see, nice vector. Malaria is a vector-borne infectious disease caused by protozoan parasites. It is widespread in tropical and some subtropical regions, including parts of the Americas, Asia, and Africa. Main parasites, and remember for yourself, and on exam you will have such questions. What are the agents, causative agents of malaria? It's not Anopheles mosquito. No, mosquitoes not flowing in your blood. Only parasites, only Plasmodium species, Plasmodia species are the main causative agents. Yes, only Plasmodium causes the disease, not Anopheles. Anopheles just mosquitoes, just transmitter, the vector. Malaria is caused by an infection, one of 4 single-celled Plasmodia species. They are Falciparum, Vivax, Malaria, and Ovale. And one more, we will remember, but the most dangerous of these 4 is Falciparum. And new species, a 5th species, Plasmodium knowlesi, causes malaria in macaques, but also can infect humans. It's like new proof that new species appear, yes, that can infect humans. Now here you can see main malaria key affected areas with chloroquine resistance. No, it's interesting map, but maybe for next slides later. Falciparum accounts for 90% of deaths due to malaria. It's the most dangerous and severe, but it's not the most spreading. Vivax. Vivax is the most widely spread species because it exists in both temperate and tropical climates. What exactly Vivax can stay even during the lower temperatures than 16 degrees in average. And now we will discuss, because without the understanding of life cycle of Plasmodium as in mosquito as in human, we will never understand the pathogenesis, all symptoms, all manifestations, all possibilities of transmission, and especially prevention. Step by step, now we will speak about all these processes, and we will come back to this picture and another one, yes, it's present in CDC, but maybe even not this, a lot of different names, but all present. Human cycle. Let's start from human cycle, because for us it's more important, yes, but believe me, without mosquito cycle we also will not understand all problems. Pre-erythrocytic schizogony or schizogony, maybe better to say schizogony. It will be more in Latin, schizogony, or in other words, tissue phase that be present in liver as a rule. No. Erythrocytic schizogony in blood, gametogony also as a rule in blood, and exoerythrocytic schizogony will be in mosquitoes. No, it will be already like the sexual way of cycle of development. Events in humans start with bite of the mosquito. You see, yes, bite of the mosquito. But remember, man is an intermediate host. A mosquito is really definitive host, because only in mosquitoes all plasmodiums make the offsprings. That's why it's like their home. Just in humans, just some cycles of development. Sporozoids are infective forms. What here you can see sporozoids, yes, that with saliva of mosquito enter into the human. Present in the salivary gland of female Anopheles mosquito. It's female, yes, remember, because just female needs the blood. Mosquito female. After bite of infected mosquitoes, sporozoids are introduced into blood circulation. You see. Period of pre-erythrocytic or liver or tissue cycle. During Plasmodium vivax, it will last 8 days. Falciparum, 6 days, yes, more quickly. Malaria, longer, yes, 16 days, and ovale about 9, like about vivax, 8-9 days. On maturation, liver cells rupture, liberate merozoites into blood system. Now we will speak more closely. Sporozoids undergo developmental phase in the liver cell. You see, yes, all sporozoids, yes, and they quickly with blood quickly move to the liver. Sporozoids are elongated and spindle shaped, become rounded inside the liver parenchyma. Multiple nuclear division develop to schizont. You see, yes, schizont. A schizont contains about 20-50,000 merozoids in liver, don't forget, in liver. because in erythrocytes also will be merozoites, but it will be not so many. Exactly in liver cells, so many merozoites present in one schizont inside the tissue phase. Exoerythrocytic, exactly this liver phase. Plasmodium, malaria, falciparum, and no-lysis sporozoites do not form hypnozoites. Remember, just ovale and vivax, okay, later, and develop directly into pre-erythrocytic schizonts in the liver. Pre-erythrocytic schizogony takes 16 days of post-infection. Schizonts rupture, releasing merozoites, which invade red blood cells in liver. Exoerythrocytic schizogony, some sporozoites do not undergo sporogony in the first instance, but go into resting stage called as hypnozoites or hibernation, and they can stay within even 2 years, reactivate to form schizonts, release merozoites, and attack red cells and produce relapses. Only for ovale and vivax its character, and they can stay even is described in literature about 5 years after first infection by plasmoids, and can form hypnozoid form, like dormant form inside the liver cells, and can stay without any symptoms, and only under some maybe exo influences, maybe even endogenic influences, can again be reactivated and lead to malaria. That's why remember that if your patient will tell you that maybe 3 years ago he or she was in tropical countries, don't exclude such possibilities of malaria. Erythrocyte cycle. After exoerythrocytic or tissue phase, we come to the erythrocytic already phase. Merozoites released invade red cells. Plasmodium vivax infects, as a rule, young erythrocytes. You will see in such interesting cartoon movie what exactly plasmodiums, in fact, what erythrocytes, it's interesting, will be. Now just try to remember. Plasmodium malaria infects all erythrocytes. Falciparum and no-lysis, as a rule, infect red blood cells of all ages. The merozoites are pear-shaped, 1-5 microns in length. The receptors for merozoites are on red cells in the glycoprotein. What here you can see how will be all these processes in the red blood cells. Liberated merozoites penetrate red blood cells. What you can see here, it's like the merozoites from liver cells, all this. It's like the first step. 4 stages occur after that. First ring stage will be, and it looks like really like a ring under the microscope. Then will be trophozoid, big like spherical shape inside the red blood cell, and already formed schizont with a lot of merozoites inside. It can be from 12 to 24 merozoites inside exactly this schizont in red blood cells, and then again rupture, and again can already such merozoites can infect red blood cells. Ruptured red cells released again merozoid, which attacked new red cells, and exactly with each this moments, each the cycles releasing of merozoid will be again new symptomatic attack or malarial attack will be of this, like malaria as a rule manifestation of malaria will be with malarial attacks, with increasing of temperature and so on, exactly with this releasing of merozoites and invading new red blood cells will be at exactly this beginning of the new attack. Continue with schizogony, you see, repeated cycles will continue. In Plasmodium falciparum infected erythrocytes with schizont aggregate in the capillaries of brain and another internal organs and can lead to very serious duration. Also, you will see a little later what complications can be. Only ring forms are seen in the blood smears, not like this. Trophozoites. After invasion, grow and feed on hemoglobin. Blue cytoplasm and red nucleus called a sign ring appearance, hence called ring form. You will see such ring forms under the microscope. Schizont. When the trophozoite is fully developed, becomes compact. Malarial pigments are scattered through the cytoplasm. The nucleus is large and lies at the periphery, starts dividing, becomes schizont. What here you can see, we will speak exactly about different types, species of Plasmodium. Plasmodium vivax. What we will see? Number of merozoites from 12 to 24. Here you can see, yes, already mature schizont. What? Imagine that it rupture and all merozoites again will infect the red cells. Red blood cells will become enlarged, you see. It's normal, but what here? Enlarge. Schaffner's dots present. What is this? Yes, yellowish brown fine granules. Schizont 9-10 microns fills and enlarged red cell. Hematocytes. But here you can see developing hematocytes. Spherical or globular. Size much larger than red cell. You see, yes, here red cell. Here will be female and male hematocyte. Falciparum. Red blood cell is normal in size. You see what all this about all development of schizont will be the normal size of red blood cell. Maurer's dots, 9 larger red spots, sometimes basophilic stipin. What here you can see, yes, it's not the Schaffner dots like here. Like a little another, like basophilic stipin. Dark brown or blackish 1 or 2 solid blocks. Hematocytes already, here you see, yes, here, this, better to see, the Maurer dots, yellow. Hematocytes, crest-centric and looks like falciparum, yes, falcix, or like half moon, or like banana shape. What exactly due to such form of hematocyte is the name falciparum. Larger than a red cell, male and female, you see 9, 10 or 12, 14 microns. What exactly such form of Plasmodium hematocyte will lead to such name falciparum. Red blood cells during malaria also in normal size contain dark brown, what you can see here, yes, dark brown coarse granules. Schizont 6-7 microns almost fills a normal sized red cells. Here you see, here, mature schizont. Contain Timmans stippling also with this yellow. Hematocytes spherical or globular, size much larger than red cells. No, almost full, all all the cells already not red cells, already like place of living for plasmodiums. And ovale. Infected red blood cells, slightly larger, contain again Schaffner's dots like vivax. Because of vivax and ovale, they are very close in development. What here you can see, yes, these dots. Schizont, 6.2 microns, fills 3/4 of, yes, you see it here already, 3/4 of the cell. Helmetocyte, spherical or globular, much larger than a red cell. Haematogony. Merozoites differentiate into male and female haematocytes. Macrohaematocytes, also called female haematocytes, and microhaematocyte, also called as male haematocyte. They develop in the red cells. Found in the peripheral blood smears, microhaematocytes of all species are similar in size. Macrohaematocytes are larger in size. What you can see here. Like vivax ovale, malaria as a rule, and falciparum. And mosquito cycle. Don't forget that in mosquito will appear new like offspring of plasmodiums. Here parasites suck up, yes, and okay, sexual cycle will be initiated in the humans by the formation of haematocyte, develop further in the female Anopheles mosquito. Only mature sexual forms are capable of further development in mosquito. In midgut, one microhaematocyte develops into 4-8 thread-like filamentous structures named microhaemats. From one macrohaematocyte, only one macrohaemat is formed. Fertilization occurs when a microhaematocyte penetrates into macrohaematocyte. It's understanding, yes? Not here. Fertilization macrohaematocyte is known as zygote. Zygote mature into ookinete, ookinete to oocyst. Oocyst already developed in gut wall. Oocyst matures with large number of sporozoites, a few hundred to thousands. Oocyst ruptures and release sporozoid in the body cavity of mosquito. You see here, yes. And it will rupture and sporozoids will migrate. There is a specific predilection for salivary glands. Now, this sporozoid is capable to transmit the infection to new host. And again, no, it's not the same. Yes, host will be, it's another, but the same like skin. Pathology and pathogenesis. Sporozoids result from sexual and sporogenic cycle of development in mosquitoes and injected into human blood serum. Events start with bite of infected Anopheles mosquitoes. Sporozoids enter liver in 1 hour, infect the parenchymal cell. And again, like start the new full cycle of development. Incubation periods varies according to species, which includes exoerythrocytic cycle time and 1 or 2 erythrocytic cycles. Remember, not from the beginning, during erythrocytic cycle, will start hematocytes formation. No, it needs like 2, 3, sometimes even 5 cycles of inside the erythrocytes for producing hematocytes. Plasmodium vivax and falciparum 10-15 days will need, can vary from weeks to months. Malaria infection can start after 28 days because exactly, no, and remember it will be such a duration of like incubation period. It's not incubation because inside the organism everything is like already in very active processes, but the symptoms, clinical manifestation not start yet. But with rupturing of erythrocytes and releasing merozoites, the clinical features, clinical manifestation will start. You see, to cycle of events in relation to red blood cells. How malaria present clinically? Stage 1, chills for 15 minutes to 1 hour, about can vary, you understand, caused due to rupture of the host red cells, present with nausea, vomiting, headache, and chills, increasing the temperature, I hope. Stage 2, fever may reach up to 40 degrees, may last for several hours, starts invading new red cells. Yes, you remember. Stage 3, patient starts sweating, finishes the episode. Cycles are frequently asynchronous. Paroxysm occur every 48 or 72 hours. In Plasmodium malaria, pyrexia may last for 8 hours or more, and temperature may exceed 41 degrees. That's why exactly it's not very often, yes, like 72 hours, but can be very long duration of exactly time of pyrexia. The most common symptoms: fever, chills, sweats, headache, nausea and vomiting, body aches, and general malaise. Uncomplicated. The classical but rarely observed, yes, malaria attack lasts 6-10 hours. It consists of a cold stage, sensation of cold, shivering, a hot one moment. As here present like funny pictures, yes, we know how to speak about so dangerous disease like without jokes. The cold stage, yes, feeling cold, shivering. You see, the person put on everything that's present in the room on him or herself. The second, hot stage, fever, headaches, vomiting, seizures in young children can be, yes, you see here, the hot stage. Put off everything, open all windows, take ventilators, and still feel hot. And the sweating stage, finally, a sweating stage, sweats, return to normal temperature, but very tired, exhausted. You see, yes, such sweating with even with sweats on the floor. Sweat. Main clinical manifestation of malaria: fever, sweating, anemia. Yes, sure, will be splenomegaly, enlarged spleen, because exactly in spleen all this infected erythrocyte are destroyed. Irritability, sure, yes. Coma can be malarial coma, retinal hemorrhage, Algid malaria, shock-like syndrome, respiratory distress syndrome. Periodicity, don't forget about such possibility, but really it's very hard to understand this periodicity. But if we will have the clear what exactly vivax or vale, yes, the periodicity will be 48 hours, and we will name this malaria. You understand, yes? Each attack, what it's the temperature curves, each attack will be on the third day. That's why malaria tertiana, the third day, tertiana. Malaria quartana, caused by Plasmodium malaria, each 72 hours, each fourth day. That's why quartana, each fourth day. and malaria tropica or malaria malignant malaria or Plasmodium falciparum malaria, and no-lazy as a rule can have 24 hours of periodicity, will be irregular with high fever, and you see that's why it's malignant malaria without any periodicity. Such severe manifestations of malaria can be that character as a rule for children younger than 5, for pregnant women can be cerebral malaria, you see, yes, high fatality rate, severe anemia, and renal failure. And also can be a lot of another, you see, hepatic dysfunction, hypoglycemia, shock, pulmonary edema or respiratory distress syndrome, fluid electrolyte imbalance, vomiting, diarrhea, secondary bacterial infection, bleeding even can be, and clotting disturbance, hemoglobinuria, or another words, black water fever, we will speak a little later. Cerebral malaria, yes, malignant malaria or falciparum infection can affect the brain and the rest of the central nervous system. It is characterized by changes in the level of consciousness, convulsion, and paralysis. Present with hyperpyrexia, oh yeah, sure, can lead to coma, paralysis, and other complications. Brain appear congested. How long malaria infection can last in men? Without treatment, Plasmodium falciparum will terminate in less than 1 year. Without treatment, can lead to death, yes. But in Plasmodium vivax and ovale persist as hypnozoids after the parasites have disappeared from blood. But hypnozoids, I hope you remember, can stay inside the organism even till 5 years. Can produce periodic relapses up to 5 years, you see. In Plasmodium malaria may last for 40 years because it's very interesting fact. It was documentary fact when the persons that live in tropical regions move in like north countries, as I remember is Canada, in Canada, and never go back home, never also was in tropical countries, and in 40 years was something like relapse of Plasmodium malaria infection. It was very interesting, yes, moment case, and call as recrudescence or X. Yes, you see X relapse. Still, we don't know such possibility. It's strange. Parasites surviving or erythrocyte or in liver cells, nobody knows. But also, sometimes if person has very serious fever without any understanding reasons, you have to investigate, yes, to make analysis also on malaria, without even epidemiological analysis. Why falciparum infections are dangerous? Can produce fatal complications: cerebral malaria, malarial hyperpyrexia, gastrointestinal disorders, algid malaria, and black water fever can lead to death. What is black water fever? In malignant malaria, a large number of the red blood corpuscles are destroyed. Hemoglobin from the blood cells is excreted in the urine, which therefore is dark and almost the color of cola. Better to say even like red-black. That's why it's very dangerous and looks like not good. It is a manifestation of infection with Plasmodium falciparum occurring in persons who have been previously infected and had been treated with inadequate dose of quinine. It is characterized by intravascular hemolysis, fever, and hemoglobin-uria. Cardiovascular collapse and shock can be abnormalities in blood coagulation and thrombocytopenia and decrease in blood platelets. Yes, malaria is a major health problem in tropical countries. Pernicious malaria, what does it mean? Is a life-threatening complication in acute falciparum malaria. It is due to heavy parasitation. Manifest with cerebral malaria. It presents with hyperpyrexia, coma, and paralysis. Brain is congested. Algid malaria presents with clammy skin leading to peripheral circulatory failure. Pulmonary edema, fluid build-up in the lungs, or acute respiratory distress syndrome, which may occur even after the parasite counts have decreased in response to treatment. Abnormalities in blood coagulation and thrombocytopenia, decrease in blood platelets, cardiovascular collapse and shock. What else can be? Acute kidney failure, yes, also due to the black water fever. Hyperparasitemia, when more than 5% of the red blood cells are infected by malaria parasites. Yes, it's not good prognosis if more than 5% are infected. Can lead to death. Metabolic acidosis, excessive acidity in the blood and tissue fluids. Yes, metabolic acidosis, it's very serious problem and can lead to the disseminated intravascular coagulopathy, to the failure of all organs and to death. So often associated with hypoglycemia. And laboratory diagnosis. Yes, how we can prove that it's really this infection and what exactly the species, because we have to know what treatment we have prescribed. Diagnostic tools, blood film examination, serology, yes, can be, and PCR investigation. Here you can see, yes, how we will make all steps of taking the blood thick or thin blood smears. What here you can see, it's making the thin smear. Here thick, yes, like drop of the blood. It looks like this. Thin and thick. Malaria parasites can be identified by examining under the microscope a drop of the patient's blood spread out as a blood smear on a microscope slide. Prior to examination, the specimen is stained, yes, most often with Giemsa stain, to give to the parasites a distinctive appearance. Sure, understanding. This technique remains the gold standard for laboratory confirmation of malaria. Thick and thin smears, the gold standard, and under the microscope you will try to find what such, for falciparum, you will try to find some of such red blood cells. Also, antigen detection methods are rapid and precise. Various test kits are available to detect antigens derived from malaria parasite. Such immunochromatographic tests most often use a dipstick or cassette format and provide results in 2 to 15 minutes. This rapid diagnostic test, RDT, yes, offer a useful alternative to microscopy in situations where reliable microscope diagnosis is not available. Malaria RDTs are currently used in some clinical settings. Also, serology detects antibodies against malaria parasites using the indirect immunofluorescence or enzyme-linked ELISA. Past, it will not have a real diagnosis of acute infection in like endemic regions. That's why still now the smears are the gold standard microscopy. Sure, the molecular like PCR investigation also possible, but still expensive and easier to use microscope. But sure, we will need the skilled laboratory staff we will need. And what this movie that I already told you about, some minutes, not long, 10 minutes as I remember. And after that we'll speak about treatment.
Learning medicine is hard work. Osmosis makes it easy. It takes your lectures and notes to create a personalized study plan with exclusive videos, practice questions, and malaria is an infection that can be caused by a few different types of Plasmodium species, which are single-celled parasites that get spread around by mosquitoes. Once the Plasmodium gets into the bloodstream, it starts to infect and destroy mainly liver cells and red blood cells, which causes a variety of symptoms and sometimes even death. Malaria is a serious global health problem that affects millions of people, particularly young children under the age of 5, pregnant women, patients with other health conditions like HIV and AIDS, and travelers who've had no prior exposure to malaria. Tropical and subtropical regions are hit the hardest. Together, the most affected regions form the malaria belt, which is a broad band around the equator that includes much of Latin America, Sub-Saharan Africa, South Asia, and Southeast Asia. There are hundreds of types of Plasmodium species, but only 5 cause malarial disease in humans, and those are Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, Plasmodium ovale, and Plasmodium knowlesi. Plasmodium vivax uses a specific erythrocyte surface receptor called the Duffy antigen. And some individuals, particularly those with sickle cell anemia, lack this receptor, meaning that Plasmodium vivax can't get into their cells. In other words, having sickle cell anemia is genetically related to having relative protection from Plasmodium vivax. Other diseases like thalassemia and G6PD deficiency make the parasite-infected erythrocyte more susceptible to dying from oxidative stress. So, despite the obvious downside to having any of these diseases, they do offer an upside when it comes to warding off a malaria infection. In fact, because malaria has historically circulated in Africa, the genes underlying these diseases are thought to have conferred a natural selection advantage and therefore become more common in the genetic pool. Now, malaria starts when a Plasmodium-infected female Anopheles mosquito hunts for a blood meal in the evening and through the night. Like a tiny flying vampire, the mosquito is drawn to carbon dioxide that gets breathed out, as well as bodily smells, like foot odor. At this point, the Plasmodium is in a stage of development, called a sporozoite, waiting patiently in the mosquito's salivary gland. When the mosquito pierces a person's skin with its long and needle-shaped tusk, called a proboscis, the tiny worm-like sporozoites spill out of the mosquito's saliva and make it into the bloodstream. Within minutes, the sporozoites reach the liver and mount an attack on the hepatic parenchymal cells, where they start asexual reproduction, also known as schizogony. At this point, the Plasmodium species vary a bit. Over the next 1 to 2 weeks, Plasmodium falciparum, Plasmodium malariae, and Plasmodium knowlesi sporozoites multiply asexually and mature into merozoites, while host hepatic parenchymal cells die. In contrast, over the next few months to years, Plasmodium vivax and Plasmodium ovale sporozoites enter into a dormant hepatic phase. where they're called hypnozoites. Hypnozoites don't divide. Instead, they snooze for a period of time before entering the process of schizogony, causing a long delay between the initial infection and symptoms from the disease. This is called the exoerythrocytic phase, because it happens outside of the erythrocyte, or red blood cell, and it's generally asymptomatic. The merozoites are then released into the blood, and each one binds to a surface receptor and invades a red blood cell. Plasmodium ovale and Plasmodium falciparum invade red blood cells of all ages, whereas Plasmodium vivax prefers to invade reticulocytes, which are young, immature red blood cells. And Plasmodium malariae and Plasmodium knowlesi prefer to invade older red blood cells. Once inside the red blood cell, the merozoite undergoes asexual reproduction and a series of transformational changes. This phase is known as the erythrocytic phase of malaria, because it happens inside of the red blood cell and generally lasts 2 to 3 days. In the first stage of the erythrocytic phase, the merozoite looks like a tiny ring within the red blood cell and is called an early trophozoite, or a ring form. In the second stage, the ring-formed trophozoite grows and is referred to as a late trophozoite. In the third and final stage, the parasite grows some more by digesting hemoglobin and leaves behind hemozoin, which under a microscope looks a little like a brown feces smudge on a red blood cell. And at this point, the parasite's called a schizont. This is the actual replicative phase, in which the parasite undergoes mitosis and differentiates into lots of merozoites, which can get released into the blood. Now, instead of going into the erythrocytic phase again, some of the merozoites undergo gametoogony, which is where they divide and give rise to gametocytes, which are little sausage-shaped sexual forms that can be either male or female. These gametocytes remain inside of a red blood cell and can get sucked up by another female Anopheles mosquito that might take a blood meal from the infected person. The gametocytes can then reach the mosquito's gut, where they mature a bit more and then fuse together to form a zygote. This part of the Plasmodium life cycle is called sporogony, and it's sexual reproduction, as opposed to schizogony, or asexual reproduction, that happened in the liver and red blood cells. The zygote then goes on to develop further, and it becomes an ookinete, and then an oocyst that ruptures in the mosquito's gut, releasing thousands of sporozoites which navigate their way into the mosquito's salivary gland, in order to repeat the cycle all over again. Now, the incubation time, which is the period of time between infection and symptom onset, varies depending on the Plasmodium species. Plasmodium falciparum incubates for a few days, whereas Plasmodium malariae incubates for a few weeks. The release of tumor necrosis factor alpha and other inflammatory cytokines causes fevers that typically happens in paroxysms, or short bursts, and correspond to the rupture of the infected red blood cells, which happens in waves of reproductive cycles unique for each Plasmodium species. For Plasmodium malariae, fevers happen every 72 hours, and is called a quartan fever. For Plasmodium vivax and Plasmodium ovale, fevers happen every 48 hours, and these are called tertian fevers. For Plasmodium knowlesi, the fever happens every 24 hours, and for Plasmodium falciparum, the pattern can vary, sometimes following the pattern of a tertian fever, while other times the fevers happen daily, earning it the name malignant tertian fever. In addition to fevers, the hemolytic anemia, which is the destruction of red blood cells, also causes symptoms like extreme fatigue, headaches, jaundice, and splenomegaly. Most Plasmodium infections have a mild course of symptoms and are generally regarded as uncomplicated malarial infections. Out of all the Plasmodium species, though, Plasmodium falciparum is known for causing the worst infections. Most Plasmodium-infected red blood cells get screened and destroyed by the spleen. Plasmodium falciparum, though, avoids this fate by generating a sticky protein that coats the surface of the infected red blood cells, and these look like knobs or little bumps. This protein causes the red blood cells to clump together and jam up tiny blood vessels, a process known as cytoadherence. This literally blocks the flow of blood so that infected cells aren't able to flow into the spleen, and it also blocks blood flow from reaching other vital organs, which can wreak havoc on them. Between hemolytic anemia and ischemic damage from blocked blood flow, organ failure can set in pretty quickly. When the brain's affected, it's termed cerebral malaria, and it results in altered mental status, seizures, and coma. When the liver's affected, it's termed bilious malaria, and it results in diarrhea, vomiting, jaundice, and liver failure. Other commonly infected organs include the lungs, the kidneys, and the spleen, which taken together create a sepsis-like clinical picture that can eventually lead to death. Together, all these scenarios are called complicated malaria. Malaria is usually diagnosed with a thick blood smear that locates parasites sitting within the red blood cells, and a thin blood smear, which directly identifies the Plasmodium species. It's also important to know the percentage of red blood cells infected by a parasite, because patients with greater than 5% parasitemia can have worse outcomes. Some common lab findings include thrombocytopenia, which is a low platelet count, elevated lactate dehydrogenase levels due to hemolysis, and a normochromic, normocytic type of anemia, meaning that the red blood cells are few in number, but those that remain are of normal size and color. Treatment for malaria is generally divided into the different stages of infection. Suppressive treatment, or chemoprophylaxis, is aimed at killing sporozoites before they infect hepatocytes. So it's usually given to travelers that are headed to a country with endemic malaria. Therapeutic treatments aimed at eliminating merozoites in the erythrocytic phase, so it's usually given during an active infection. The exact medication or group of medications that are used to treat an active infection depends largely on the severity of the infection, the age and pregnancy status of the patient, the local malarial resistance pattern, which depends on the geography, and the Plasmodium species causing the infection. It's also important to not take the same medication to treat an active infection that was previously used as chemoprophylaxis. Gametocidal treatments aimed at killing gametocytes, which prevents spread of disease, and thus the creation of future resistant forms of the parasite. Lastly, radical treatments aimed at killing hypnozoites in the liver from a Plasmodium vivax and Plasmodium ovale infection. For the most part, cases of uncomplicated malaria resolve with treatment. Even after recovery, some individuals can get symptoms after a period of time, and this is called recurrent malaria, and it's broadly divided into 3 underlying causes: recrudescence, relapse, and reinfection. Recrudescence refers to ineffective treatment that didn't completely clear the infection, a problem common when there are high rates of anti-malarial resistance. Relapse refers to situations where the blood was cleared of merozoites, but hypnozoites persisted in the liver, and then emerged to cause more problems. And reinfection is when an individual was effectively treated, but a completely new infection caused a new bout of malaria, a problem common in endemic areas, since a single infection doesn't make a person immune to malaria. Instead, there's an acquired ability to tolerate Plasmodium infections, which relates to the degree of exposure to a variety of different strains. Since malaria is spread by mosquitoes, anything that prevents mosquito bites can help, like full-body clothing, mosquito repellent, sleeping in insecticide-covered mosquito nets, and using indoor insecticide sprays. In addition, Anopheles mosquitoes like to lay their eggs in small, shallow collections of fresh water, like containers sitting outdoors during the rainy season in tropical countries. To control the mosquito population, it's important to empty out these containers and any other stagnant collections of water. Finally, an emerging vaccine against malaria is currently being studied in clinical trials, and it's called, brace yourself, RTS-S/AS01 vaccine. I'm guessing malaria be gone was already taken. All jokes aside, though, this vaccine consists of a recombinant fusion protein which targets an antigen found in the P. falciparum membrane. And while more clinical trial results are necessary before recommending widespread employment of the vaccine, it looks really promising. All right, as a quick recap, malaria is a life-threatening mosquito-transmitted infection caused by Plasmodium parasites in which the Plasmodium feeds and grows inside hepatocytes and red blood cells. Symptoms are primarily caused by the rupture of red blood cells that usually result in high-grade fever paroxysms that improve over time, but can occasionally cause severe complications and death. Thanks for watching.
Okay. I hope you enjoy this video, and it will help us to remember all these processes. Maybe by watching is more understandable. But treatment. Here I propose all possible drugs. No, maybe not all, but the most common, yes, and the most useful here for regions chloroquine and hydroxychloroquine, for regions where the Plasmodium falciparum is sensitive, and for all another species. Here mefloquine and atovaquone provanil for regions where falciparum is resistant to chloroquine resistant. You understand Primaquine for dormant forms, but not for pregnant women. Quinine, doxycycline, all another can be used during all forms, any forms of the infection. In endemic areas, the World Health Organization recommends that treatment has been started within 24 hours after the first symptoms appear. Treatment of patients with uncomplicated malaria can be conducted on an ambulatory basis without hospitalization, but patients with severe should be hospitalized as quickly as possible. What does it mean, presumptive treatment of malaria? Presumption. In an area with high transmission of malaria, it should be presumed that all cases of fever are due to malaria. It doesn't matter. Fever means malaria. Treatment. First loading dose of chloroquine should be administered immediately after collecting the blood specimen, even without waiting for its report. If the fever is indeed malaria, this treatment alleviates symptoms early, maybe well before the test result is available. If it is malaria, chloroquine also prevents the spread of malaria by destroying the hematocytes of Plasmodium vivax, the most common malaria. If it is not malaria, nothing is lost. For chloroquine at this dose is safe and has no adverse effects. Radical treatment is administration of Primaquine to all confirmed cases of malaria because it stops the hematocyte producing. In Plasmodium vivax malaria, 2 weeks therapy with Primaquine completely cures the infection in the host by its tissue schizonticidal, like killing activity, and thereby prevents relapses. In Plasmodium falciparum malaria, a single dose of Primaquine destroys the hematocytes, thereby prevents the spread of the infection into the new mosquito. Primaquine is active against the dormant parasite liver forms hypnozoid and prevent relapses. Primaquine, but what is the most not good and dangerous but important, yes, not be taken by pregnant women or by people who are deficient in glucose 6-phosphate dehydrogenase, G6PD. Patients should not take Primaquine until a screening test has excluded G6PD deficiency. Drug resistance, yes, it's very dangerous situation, but Plasmodium falciparum already all over the world produce the resistance. Chloroquine-resistant Plasmodium falciparum, you see CRPF abbreviation, first developed independently in 3 to 4 foci in Southeast Asia, Oceania and South America in the late 1950s and early 60s. Since then, chloroquine resistance has spread to nearly all areas of the world where falciparum malaria is transmitted. Here also you will see something like cartoon movies of appearance of this resistance. Yes, 60s, 70s, 80s, 2000. You see, almost all regions of this malarial belt. And here you can see the also resistant to mefloquine, of chloroquine of Plasmodium vivax resistance. And now I want just some words about also vaccination and prevention after this shot. It's not short, but it's I think it's really needed, this also like cartoon movie, because it will explain the influence of each drug on the Plasmodium, and maybe it's a little complicated, but I hope it will help you.
Treatment of malaria, basically the pharmacology. So, of course, a vaccine would be optimal to prevent people from getting malaria, but developing a vaccine has a lot of difficulties, cost, and I guess the different strains of the Plasmodium species, and I guess the ever-evolving mutations in the genetic material also plays a role. So before getting into the treatment of malaria, let us again recap the life cycle, because treatment targets different stages of the parasite's life cycle. The parasite responsible for malaria are the Plasmodium parasites. The Plasmodium parasite is carried by a female Anopheles mosquito. The female Anopheles mosquito is infected with the parasite. The Anopheles mosquito carries the parasite in its salivary glands as sporozoites. The sporozoites is a form the parasite is in when it enters the human body. During feeding, the mosquito will expose the sporozoites to human circulation. The sporozoites will travel to the liver, where it will enter the hepatocytes. Within the hepatocytes, the sporozoites will replicate, mature into schizont, and finally rupturing, releasing many merozoites, which is another form of the parasite. And these can be called the daughter parasites, which again have to mature. So the merozoites then enters the human circulation, the blood. But just going back to the sporozoites, the different Plasmodium species, right, they have different abilities. And there are 4 or 5 main Plasmodium species. So, for example, the Plasmodium ovale and vivax sporozoites are able, they have the ability to remain dormant in the hepatocytes and form what's called a hypnozoite. And after many months or years, it will then replicate and then release its merozoites into circulation. So now let's go back to the merozoites, which are in circulation. So the merozoites are bad, and they invade red blood cells. Within the red blood cells, it enters the ring. It will enter what's called the ring stage. It will then replicate to form a trophozoite and mature to become a schizont before rupturing, releasing again many merozoites. This cycle can then continue. So, you produce more merozoites, and yeah. Now, the rupturing of the red blood cells is responsible for anemia in malaria, The rupturing of the red blood cells are also coincidental with the clinical manifestations of malaria, which, as we know, are the fever, rigor, chills, tremors. Now, going back to the ring stage of the parasite's life cycle, which is in the red blood cells, right? The parasite can also enter a sexual cycle where it can become a female or male gametocyte. When a new uninfected Anopheles mosquito then comes along for feeding, it can take in these gametocytes. The gametocytes will enter the mosquito's stomach. The Anopheles mosquito is now infected with the parasite. Now, in the stomach now of the mosquito, the male and female gametocytes, which have just been taken in, can form a zygote. The zygote will mature into a eukaryote, then be released by the stomach as an oocyte. The oocyte then matures and ruptures, releasing its content, which are the sporozoites. The sporozoites, as we know, will remain in the salivary glands until the infected mosquito will feed again. It will feed off humans again, and then this whole cycle can continue. The mosquito can feed on another human, another person that is healthy. And then if this person was infected, the person will obviously be unhealthy. Now, malaria is a big problem worldwide, particularly in developing countries. In Africa, many children under age of 5 die to a malarial infection. Thus, it is a very important public health issue. Aside from prevention strategies, there is also treatment option which is prophylaxis prevention, or treatment when the symptoms arise. And we will now look at the drugs, the pharmacology drugs used for malaria, and their mode of action using this diagram we just drew. I hope this will help. So for anemia, I mean serious anemia, blood transfusion can help to replenish the loss of blood. Now let us look at the big drug class known as quinolones. To understand quinolones, we have to understand how the Plasmodium parasite survives in red blood cells. So here's a red blood cell, and here is the parasite. The parasite has what's called a vacuole, which is important for basically nutrition, metabolism. Now, the red blood cells are red blood cells. They contain many hemoglobins, right? And the hemoglobins, as we know, are responsible for delivering oxygen to body tissues. Well, the parasite within the red blood cell will use the hemoglobin. The hemoglobin is metabolized by the parasite in the vacuole, in the parasite's vacuole. It will break the hemoglobin into heme and protein. The parasite will use the protein for some stuff, but it's the heme that is important here. See, what happens is that the parasite uses heme for energy. While metabolizing the heme, the parasite actually converts heme into hematin. Now, hematin is actually a dangerous chemical, so hematin is toxic to the parasite. So what the parasite does is that it converts hematin into a non-toxic form called hemozyme. So now the drug quinolones, the drugs under quinolones, they actually target this conversion from hematin, which is toxic to the parasite, to hemozyme. So these quinolones I'm talking about are quinines, lumefantrine, chloroquine, and amdioquine. Now, I hope I pronounced these right, but, you know. So these guys, these quinolones, these guys essentially inhibit hematin to become hemozyme. Thus, if hematin is not converted, it will be very toxic because hematin is toxic to the parasite, and the parasite dies. However, there is a particular strain of the parasite called the Plasmodium falciparum, who has been able to develop resistance against chloroquine. See, what happens is that the parasite have been able to develop a pump. This pump pumps out the drug from the vacuole so that the chloroquine has no effect and the hematin can be successfully converted to hemozoin by the parasite. This resistance is known as chloroquine resistance. As the pump, it specifically removes the chloroquine drug. So those are the quinolones. Now, all these drugs are related in structure. Just have to point that out. The next antimalarial drug targets the mitochondria of the parasite. This drug is known as atovaquone. Look, the next antimalarial drug, which I will most likely mispronounce, targets the mitochondria. This drug is known as atovaquone, and it targets the cytochrome electron transport chain in the mitochondria. Thus, we can say that atovaquone inhibits the parasite from making energy. Our own bodies also have cytochromes, but this is distinctively different from the parasitic cytochrome. Side effects of atovaquone include skin rash, fever, insomnia, and nausea. Some Plasmodium parasites have actually been able to develop resistance to this drug due to mutations in their cytochrome gene. The next antimalarial drug we will talk about are the ones that target food metabolism. The parasite can use PABA, P-A-B-A, and through the enzyme DHPS can make folate. Folate can then be converted to tetrahydrofolate through the enzyme DHFR. Tetrahydrofolate can then be converted to thymidine through several reactions. Thymidine will eventually be used for the genetic material of the parasite. Thus, if we have drugs that inhibit this process, we can kill the parasite. These drugs are also antibiotics and include sulfonamides, which inhibit the enzyme DHPS, and diaminopyrimidines, which inhibit DHFR. The parasite have also been able to develop resistance to these drugs through genetic mutations of the enzyme, making it different enzymes. So going back to atovaquone inhibits cytochrome, remember? And specifically, actually, it targets the sexual life cycle of the parasite, thus inhibiting gametocyte formation. So atovaquone targets the sexual life cycle or sexual cycle of the parasite. There is another drug that targets the parasitic liver stage. So this drug I'm talking about is called primaquine. Now, primaquine is a radical cure, and it prevents the relapse of latent liver hypnozoites. So it targets Plasmodium ovale and vivax, because these are the Plasmodium strains that can become dormant. Now, primaquine is contraindicated in people who have glucose 6-phosphate deficiency because it can cause hemolytic problems. And it is also contraindicated in pregnancy, like many other drugs are. The next antimalarial drug we will look at, and it's a very important one, are known as artemisins. These guys are essentially inactive when they're given. And when they're given to people, they are activated by ferrous heme or free heme. So when activated by the heme, the artemisins, they create a carbon-centered radical, which will essentially kill the parasite. The free radicals formed kills the parasites in several ways. These free radicals will kill the parasites through damage to their lipids and vacuole membranes, or they will inactivate the Plasmodium proteins, alkylation of heme, and also it will interfere with the conversion of hematin to hemozoin. Thus, it will increase hematin in the parasite, which is toxic to the parasite. So all in all, active artemisins is toxic to the parasite. Doxycycline is another anti-malaria drug which you might remember as being an antibiotic. Doxycycline is essentially a protein synthesis inhibitor. Tetracycline, another antibiotic and protein synthesis inhibitor, is also used for anti-malarial treatment and prophylaxis. Its side effects include esophagitis and photosensitivity, and they are contraindicated in pregnancy. So now let us look at what to give to certain patients in different scenarios. So for example, we'll actually mainly focus on P. falciparum because that's the main strain of Plasmodium that is associated with morbidity and mortality in the developing countries that have high prevalence of malaria. So for uncomplicated P. falciparum malaria, we would give a patient artemisins plus a quinolone, such as, firstly, lumefantrine, or if not lumefantrine, we would give them another type of quinolone. Or if we can't give them the quinolone, we would give them artemisins plus a protein synthesis inhibitor, such as doxycycline or tetracycline. Now, for complicated P. falciparum, complicated is really when you have association of really organs, when the organs are affected, and you would give them IV artemisins or quinines. For radical cure, which is basically targeting the dormant stages of the malaria parasite, so we're targeting the ovale or vivax in the liver, we would give them primaquine. For prophylaxis, we would give a traveler, for example, chloroquine. And we would give them chloroquine for areas which there is no resistance to chloroquine. So these are areas such as Central America. But if the traveler is going to places where the malarian parasites are chloroquine resistant, we would give them amivaquone or another type of chloroquine. Anyway, I hope you enjoyed this video. Thanks.
I also hope you enjoy this video, because I hope you will remember and understand a little better how all drugs influence exactly on Plasmodium and on human body also. And for the finishing of our lecture, I want to remember that World Malaria Day is April 25, which commemorates the date is in 2000 when 44 African leaders committed to cutting malaria deaths in half by 2010. This year's World Malaria Day theme is counting malaria out. Also, what will be maybe the best years for stopping malaria will be developing the vaccine. Include pre-erythrocytic or liver stage vaccines, yes, that aim to protect against the early stage of malaria infection, blood stage, as also as all drugs influencing also the vaccine, also the same steps of development try to stop. Blood stage vaccines that aim to reduce the severity of disease, and transmission blocking vaccines that are intended to prevent mosquitoes that fed on an infected person from spreading malaria to new hosts. But here you can see, yes, like all these possibilities in the liver, pre-erythrocytic vaccine, in the bloodstream blood stage vaccine, and inside the mosquito transmission blocking vaccines. But the simplest method is to avoid the mosquito biting, yes, and to sleep with bed nets, with spraying by different insecticides or repellenters and so on. Task for you. the goal of medical humanity, malaria no more. Is it good or not? And will it help? And is it really needed? Maybe no, malaria has to stay, but we have to understand how to protect humans, yes, without being to catch very serious and to develop very serious disease. It's task for you. Maybe on practical classes we will try to discuss this situation and problem. That's all for today. Be attentive.